everyone. In today's class, we will understand the third component of the nucleotide, that is phosphoric acid. When we see the figure of phosphoric acid, it has one oxy group and it has three hydroxyl group. Oxy group is present here and hydroxyl group is present in these three sites. So this is the simple structure of phosphoric acid. Now this phosphoric acid will always be attached at the fifth carbon of the pentose sugar. If you remember, in the last class we said that a pentose sugar consists of 5 carbons. So this phosphoric acid will always be attached at the fifth carbon of the pentose sugar, which was lying outside of the sugar ring. Now, since we know all the three structures of nucleotides, that is nitrogenous bases, pentose sugar and phosphoric acid, let us try to understand the complete formation of a nucleotide. See, in this figure we have a nucleotide. This is a monomeric unit of a nucleotide. So we can find here a pentose sugar. This is sugar, and in the first carbon, first prime, 1 prime, I have written here 1 prime, there is attachment of a nitrogenous bases. If you remember the structure, this structure denotes a purine ring. It is dicyclic, if you remember from the last class. And the third component, that is the phosphoric acid that we just saw now, is attached at the fifth carbon. So this is the fifth carbon of the pentose ring, and thus gives the complete structure of nucleotide. In the other figure, there is a attachment of sugar with the pyrimidine. If you see this figure, this is a pyrimidine, and the first position of the pyrimidine is attached with the first carbon of the pentose sugar, and there is attachment of phosphoric acid also. So here also we get the complete structure of a nucleotide. Now, to form this complete structure, there are certain bonds in between these different components of nucleic acids. So let us first try to understand the glycosidic bond. So the glycosidic bond is the bond between the pentose sugar and the nitrogenous base. Glycosidic bond is the bond between the pentose sugar and the nitrogenous base. So when we see the structure of a nitrogenous base, in this case I have taken the structure of adenine. If you remember, the chemical name of adenine was 6-aminopurine. So we have a amino group in the 6th position. Now this is the 9th position. This 9th position is attached to the first carbon of the pentose sugar. So there is removal of water. You see here in the figure, the H group from the nitrogen and the OH group from the first carbon is released in the form of a water. So once the water is released, there is formation of a glycosidic bond between the nitrogenous base and the pentose sugar. Now another point is that the nitrogenous base is free to rotate around the glycosidic bond. That means this nitrogenous base that is connected to the pentose sugar can rotate freely in different conformations. So there are two possible conformations. One is syn type and the other one is anti. So the purines, they can have both syn as well as anti conformations, while pyrimidines will have only anti conformations. So what is syn and anti conformations? Let us see in the figure. So in this figure I have drawn this syn conformation. So when we say syn conformation, it is in its original conformation. You see here, as we saw in the last class, the nitrogenous base is in its original conformation. and attached to the pentose sugar. While in case of anti, there is anti-rotation, means anti-clockwise rotation of the nitrogenous base. The 9th position is here and the first cyclic moves towards the right side. While in syn conformation, the first cycle is in its original conformation. That's why it's called syn, while in the anti, it rotates anti-clockwise. So it is called as anti-conformation. Thank you.